Good afternoon. Thank you for the opportunity to present my work entitled Data-Driven Refinement of Gene Signatures for Enrichment Analysis and Cell State Characterization. Gene set enrichment analysis, or GSEA, is a community standard method for testing the activation of pathways and processes in gene expression data. Briefly, GSEA works by ranking genes in a gene expression matrix by their correlation with sample class labels and testing if the members of an a priori defined gene set are overrepresented at the top or bottom of the rank list. This is done by computing a running sum over the rank list that is incremented if the gene is in the gene set and decremented otherwise. The maximum deviation from zero, either positive or negative, is the enrichment score. GSEA relies on gene sets whose member genes are all coordinately up or down regulated when samples have a specific pathway or process activation or a particular phenotype. This is illustrated by the toy pathway diagram here that contains genes upregulated, downregulated, and not differentially expressed when a pathway is activated. Including all of these genes in the gene set results in a lower enrichment score. As an example, we selected all of the cell lines from the cancer dependency map for which the protein abundance of HER2 was available. We then computed the enrichment scores for the reactome signaling by ERB2 gene set in each sample. We found only a weak correlation between the enrichment scores and the more direct measurement of the HER2 signaling phenotype, indicating that this gene set contains member genes that are not coordinately regulated. Gene sets can lack this property of coordinate regulation for several reasons, including curation decisions and deriving a gene set based on a single experimental context. We first attempted to refine gene sets by generating the molecular signatures database hallmark collection. This reduced redundancy, but issues of context specificity and coordinate regulation remained. In recent years, the amount of publicly available multi-omics compendia has increased, so we asked if we could leverage these large-scale collections to build data-driven, refined gene sets in order to improve their sensitivity and specificity in GSEA. The method we propose works with a multi-omics compendia and refines an existing gene set using two inputs, a gene expression matrix and, for the same samples, additional more direct measurements of phenotypes such as CRISPR screens or proteomics data. We then extract the gene expression profiles for the gene set genes, and this matrix is then subject to non-negative matrix factorization, a dimensionality reduction that yields two smaller matrices whose product approximates the original. We then select a direct measurement to which we would like our gene set to correspond, and compare it to the H matrix from the decomposition, which has a column representing each sample from the original gene expression matrix. The row, or component of H most associated with the direct measurement, is termed the signal component, and we then identify the genes from the original A matrix most associated with the signal component to comprise the refined gene set. In short, this method identifies lower dimensional transcriptional patterns in the expression space of the original gene set and defines new gene sets based on this coordinate regulation. For an example of this method, we will return to the reactome signaling by ERB2 gene set. We use the RPPA abundance of HER2 as the target phenotype. The top rows of this heat map are the same as on the previous slide, showing a low association between the gene set and the HER2 protein abundance. The bottom row shows the enrichment scores for the same samples, but with the refined gene set. This refinement process has extracted a component of the original gene set whose members' coordinate regulation is a more sensitive and specific indicator of the HER2 signaling phenotype. We are currently applying this method to a wide variety of gene sets and phenotypes in order to build a new refined gene set collection. We are also working on a variation on this method that will allow the extraction of multiple gene sets from an existing gene set in order to build more coordinately regulated gene sets for specific signaling contexts. I would like to acknowledge the members of my lab, our collaborators, and my funding sources, and would be happy to take any questions via my email here. Thank you for your time.